Now the next phase is packaging. The third phase is packaging. Now the pairing of homologous chromosome is completed in packaging, and chromosomes, they become more and more thick. Pairing is completed. The chromosomes, they become more thick. The bivalent will have 4 chromatids, as I have told you, and they will be wrapping around one another. The non-sister chromatids of homologous chromosomes, they exchange their certain segments. Exchange of segments. This is known as chiasmata or chiasma, and the process is known as crossing over. Crossing over means certain segments of one chromosome will be transferred to another, and that chromosome will give its segment in return, and this is known as chiasmata. This joining is called chiasmata, and the process is known as crossing over. Now here the shuffling of genetic material occurs. But this is non-parental recombination, because parent cells are not involved. These are happening only in the single individual because of the change of segment or exchange of segments within the chromosome of the same individual, not because of the parents. So when it will happen at the parent level, that will be parental recombination, but at this stage it will be non-parental recombination. Now packaging will last for days, weeks, or even years. For example, in man, the packaging lasts for a few days, and in gymnosperms it lasts for few weeks or even for years. Whereas the previous stages, the leptotene or zygotene, they last for only few hours. The next phase is the diplotene. The fourth one is diplotene. Now the paired chromosomes, they are already close. Now they start to repel each other. The homologous chromosomes, they start to repel each other. And they begin to separate. However, the separation is not completed. The incomplete separation. Because homologous chromosomes, they remain united or combined at certain points of chiasmata or the point of interchange, the rest parts, they repel each other, but only the points of interchange or the points of chiasmata, they are attached to one another. So each bivalent has at least one such point, otherwise they will be separated. Now here the synaptonemal complex will be disappearing, and chiasmata becomes more and more visible, because as chromosomes will move apart, their points of attachment will become more and more prominent. The last stage is diakinesis. During this phase, the condensation of chromosomes reaches to its maximum. There will be maximum condensation. At the same time, separation of the homologous chromosomes will be completed, but they are still joined at the point of chiasmata. Separation is completed, but they are still attached at chiasmata. Now at this point the nucleoli, they also disappear. Now what is the importance of recombination? Recombination helps us to understand the genetic map, the mutations that are occurring, and the proportion of parental and non-parental recombination. So crossing over is important with respect to these points. So this is all about the prophase 1 of meiosis 1. In the coming lectures we will discuss about the other phases. Till then, take care of yourself. If you have any questions, you can ask in the comment section. Stay tuned, stay connected, keep studying, take care of yourself. Allah Hafiz.